much. I have first a quick announcement to do about retrocrowds. Please, trainees, students, postdocs, get courageous and ask PIs questions. You can, if you don't know how to start, you could ask, why can we not park on Bunktown Road? If you don't get the jokes, this is a great opening, and ask the people why. This is incredibly funny. So, but any other question is also acceptable. So this is meant to basically provide an excuse to start talking to people that you don't know. So, and the cards are really cute, and you find some really interesting data on the other side, but you need to get your hands on those cards. Otherwise, you will not know. So, I hope, if you have any questions about this, Andres and I can provide you with additional questions. So, with no more, so it's a real pleasure to be here. Welcome everyone to the first keynote speech of the meeting. It's really a great honor to introduce Professor Jaquelin Dudley. She's a professor of molecular biosciences at the University of Texas in Austin. After receiving her PhD from the Baylor College of Medicine, Jackie did her postdoctoral training in the lab of Harold Varmus. She joined then the faculty at the University of Texas at Austin in 1983, and the rest is history. She has built an amazing independent group that really has taught us a lot about the pathogenesis and the mechanisms of action of retroviruses. She's both a retrovirologist and also a breast cancer biologist. Her work has really shaped our understanding of proto-oncogenes, how they are amplified, and the mechanisms by which MMTV induces breast cancer. Her group has also discovered that MMTV encodes a Ref-like protein which degrades activation-induced deaminase as well as the ApoVec proteins. Last but not least, her lab has been working on MMTV-based viral vectors for gene therapy. Jackie has published over 86 papers. She's an elected fellow of the American Academy of Sciences, and she wrote some of my favorite ApoVec reviews, I have to admit. So she serves on editorial boards of several of our most favorite top virology journals. But she's also a dedicated teacher and an inspiring mentor. She's a fearless trailblazer, as you might know, and she is a fantastic role model for many, including myself. She has been a big supporter of Cold Spring Harbor. She told me earlier that she has been here since 1981, so that makes it more than 40 years. So with no further ado, I'm really looking forward to hearing your story. Thank you so much for sharing.
Undo there. There we go. Well, thank you so much to the organizers for inviting me. It is really my honor to speak to this group of retrovirologists. So I decided today, and I guess there was a little discussion earlier in the meeting about the difference between a simple retrovirus and a complex retrovirus. So I decided to have, since I've been talking about this for a while, to give you some simple lessons that we've learned from another complex retrovirus. So since this is sort of a perspective on my career, I thought I would start out by, many of you probably don't know what the origin of mouse memory tumor virus is. And so this really dates back to the origin of inbred mice. And when there were these original strains were derived, they noticed that there were some inbred strains that developed high memory cancer and some that were low memory cancer. And what they rapidly found out was that if you foster nursed a low cancer progeny on a high cancer female, then those animals got essentially a 90% tumor incidence. And the reciprocal cross indicated that the cancer incidence really reflected the cancer incidence of the mother. And then subsequently, John Bittner really initially defined that there was a milk agent that was transmitted through the milk. This was the summary of the original Bittner report in Science in 1942, and it was called the Bittner agent and later mouse memory tumor virus, or MMTV. And essentially what he concluded that, following the centrifugation and in the supernatant fluid, it's possible that the active agent is a colloid of high molecular weight. And so one of the characteristics that was identified originally from retroviruses was there was a classification by electron microscopy, and they were classified into A and B and C particles. And so MMTV was actually a B particle, and it was quite unique because it had this eccentric or off-center nucleoid. The report from Lyons and Moore, who also studied MMTV in Nature in 1962, was that RNA containing particles with spikes, similar to influenza virus, with this eccentric nucleoid, were consistent with extracellular B particles, because there were also intracellular ones that had been noted. So then, about a decade later, then Wade Parks, Ed Skolnick, and Kozakowski in Science reported that when they treated mouse memory tumor cells in culture, but not uninfected cells, that when they added a synthetic glucocorticoid called dexamethasone to these cell lines, that it stimulated the production of MMTV, both the RNA and the proteins, as much as 20-fold. And so these studies suggested that MMTV was actually the original hormone-regulated promoter. So it has become a paradigm for hormone-responsive gene expression. Okay, so when I was in Harold Varmus's lab, there was still the not well-defined what the messenger RNAs encoded. And so it had been reported that there was, it was assumed there would be a GAG-Pol messenger RNA like Rouse, which was sort of the prototype at the time, and that there would be an envelope messenger RNA, but what I set out to do was to see if there were other messenger RNAs, and so I ended up purifying from, it was probably 20 large roller bottles of cells, and extracting the RNA, purifying the poly-A RNA, size fractionating it, and then taking that RNA and hybridizing it to a filter, eluting that RNA, and putting it in an in vitro translation reaction. That would still be very difficult in my lab today. And so what we found out is when there was the presence of this lower molecular weight messenger RNA that was different than what was found in virion RNA, that you could immunoprecipitate from the in vitro translation products. You could IP with antibody to the surface protein known as GP52. And then later, John Majors, a friend and colleague that I first met at UCSF, when he started his own lab at WashU, he developed a method and binding assays together with EM and purified glucocorticoid receptor with cloned DNA. So these were in the first days of cloning. And he found that the purified glucocorticoid receptor bound to the ends of viral DNA, which corresponded to the LTR. And so it suggested that the mechanism for glucocorticoid responsive gene expression was the binding of the activated or hormone-bound receptor to the LTRs to drive the increase in MMTV expression. So subsequently, when I left Harold's lab, then Roelness started to look for what were the integration sites. So it was known that retroviruses integrated into DNA, and so they were looking for insertional events within mammary tumors, and what they did is they screened for mammary tumors that had only one insertion, and then by a very long method compared to today's techniques, they were able to identify a common insertion site within MMTV-induced low-copy breast cancers, and this was found to be overexpressed compared to normal mammary glands. And this gene, which they called integration site 1 or INT1, was found to be a homologue of Drosophila wingless, and then they combined the two names, and the subsequent gene was called WNT1. You can see here, there's been a huge amount of work defining the WNT pathway and its important role in beta-catenin dependent signaling. So around the time that they were looking for insertion sites in MMTV, I was actually doing a second postdoc with Rex Risser at University of Wisconsin McArdle Labs. And so Rex was really well known for the genetic characterization of murine leukemia virus, and he had a number of what he thought were B-cell lymphomas in the lab, which turned out to be T-cell lymphomas. And we knew that we could detect new insertions, and so you can also tell the genotype of what strain of mouse that you're looking at compared when you look at the endogenous virus. So one thing that we were really surprised by is that these T-cell lymphomas had additional copies of MMTV DNA in them. And those endogenous copies, and you ran them compared to MMTV-induced tumors, they had GAG-PAL and OMV messenger RNAs that were smaller, and in Judy Ball and Heidi Diegelman compared this by heteroduplex mapping and found that there was about a 400 base deletion in these T-cell derived MMTVs. So when I was also starting my own lab, I was looking for whether these T-cell lymphomas had novel insertion sites compared to the MMTV-induced mammary tumors. And you can see here a couple of papers that I published early on in my career. And you might note that I am the sole author on these papers. So I did all of the experiments, I wrote the grants, I wrote the paper. And this would not happen today. And so what I found, I can't say we, was that these T-cell tropic MMTVs showed non-random insertions, and they were statistically near transcriptionally active L1 elements. And at the time, this is where I got the NAR paper, a full-length copy of transcripts of L1s had not been reported. Whoops. Go back. Okay. So then a talented graduate student and postdoc in the lab went on to map where some of these T-cell tropic MMTVs were localized. And Judy Ball called that type B leukemogenic virus because it was isolated as a mixture, but it was an MMTV, really, related virus that was responsible for the T-cell lymphomas. And what we found is they often inserted near MYC, and often they inserted, just like in the MMTV-induced mammary tumors, upstream and in the opposite transcriptional orientation, or downstream in the same transcriptional orientation. So these studies actually showed that TBLV also uses enhancer insertion to activate proto-oncogenes, and MYC was overexpressed. I haven't shown it here. But in contrast to the MMTV enhancer, there was a novel enhancer in TBLV, and it activated transcription from a separate set of oncogenes. So the LTR specified which prototype of oncogene would be selected in that particular tumor type. And so we showed c-MYC, and then in other studies, Dana showed there was also a orphan retinoic acid receptor that was activated in these tumors. And then Jenny and Dana also wanted to know, since some of these insertions had different numbers of repeats of the enhancer, how that affected MYC expression. And we couldn't really do that with overexpressing MYC, because when we transfected the cells, and if you overexpress MYC, it killed the cells. But what they did is develop a reporter gene in which the whole locus of MYC, including 5 kb downstream and 3 kb upstream, they inserted the renylluciferase gene into exon 2, so it would report on use of the promoters of MYC without activating or having overexpression of MYC itself. And they could show, again, that just insertion of the LTR in either orientation would activate this reporter. So we also wanted to know what the nature, we knew there was a new enhancer, but there was also a deletion that was associated with these T-cell tropic viruses. And so my very first graduate student, Lillian Sue, was doing cat reporter assays where she would put a wild-type LTR or a deleted LTR upstream of chloramphenyl acetyltransferase. So these are the old reporter assays. And so what she found is that the wild-type MMTV LTR was dexamethasone inducible, but that the deleted forms of the LTR actually had higher basal expression. And so what we found was that this TBLV, not only does it have a new enhancer that drives expression in T cells, but it lacks negative regulatory elements, or NREs, to activate this, elevate this basal reporter expression. And then together with Susan Ross's lab, Susan made mice that had various LTR reporter gene fusions, and we showed that LTR transgenic mice lacking these NREs showed elevated MMTV expression in most of the tissues. And so we also did that to look at these various tissues. So here's the nature of the deleted reporter genes compared to the full-length LTR. And then we also made probes to look specifically at the transcripts. And you can see here that, the expression in the mammary gland for this truncated reporter was very high. We couldn't measure it. But it was also elevated compared to the wild-type LTR in almost all these tissues, and especially in some of the tissues that normally don't show good expression at all, like the kidney or the heart. So then another very wonderful graduate student, Jinchi Liu, took on the task of purifying the factors that bound to the negative regulatory elements. And so we had developed a reporter assay, not a reporter assay, but a gel shift assay, where we could measure the formation of two complexes that bound to the NREs. And we also didn't know what those were, but what we did was, I think, got the thymi for around 400 rats. Jinshi purified it into a single band on a gel, and then we sent it to Cold Spring Harbor so that they could do the mass spec on it. And this was Rouge Kobayashi, who was a wonderful collaborator, and he was the one that showed that the predominant band of this affinity purified protein was special AT-rich binding protein 1, or SAPB1, and there was also another protein, related homeodomain protein known as CUX1. So we originally called them ubiquitous binding protein and NRE binding protein because NBP, or SAPB1, was tissue specific and found more in T cells than in other tissues. And then Chuen took on the effort of showing that these proteins were really what was responsible for the negative regulation of MMTV in the NREs. So we actually got mice on a mixed background. Mary Lozano bred them to homozygosity on the BALB-C background, and then we infected mice that were missing the C terminus of CUX1, either the wild-type mice or heterozygotes, because the homozygote condition was lethal. And what we found is not only was MMTV expression increased in the heterozygous mice, but also milk-specific proteins such as whey acidic protein, alpha-lactalbumin, and beta-casein, but not all the casein. So epsilon casein and then also WDM1 were controls that were not. So this wasn't any gene, but it does show you that MMTV is following the expression of milk proteins because it is a milk-transmitted virus. And then Urmila sought to determine more molecularly what was the basis for the absence of negative regulation in the mammary gland. And so we knew that SAPB1 is relatively T cell specific, so it is not really expressed in the mammary gland. It's expressed in T cells, which is one of the cell types that's needed for transmission of the virus. But what Urmila did is she developed a system in tissue culture for taking undifferentiated mammary cells, and then if you cultured them in the presence of lactogenic hormones and extracellular matrix, what we saw was the development of a truncated form of CDP. So we also see that if we look in whole mammary glands over the process of differentiation, and so in pregnant mammary glands, you start to see the relief of negative suppression by developing a dominant negative form of this negative regulator of transcription. So it goes from high CUX1 expression in virgin mammary glands. In pregnant mammary gland, you get cleavage to this dominant negative, and then you get high MMTV expression and then hormone-stimulated transcription. Okay, so another story that we have worked on is that in 1981, Larry Donauer, when he was in Gordon Hager's lab, started to, he got the sequence essentially of the entire MMTV genome, and then discovered that there was an open reading frame in the LTR. And at the time, most of the LTRs did not have coding capacity. And so approximately 10 years went by before anyone realized what the function of those of that LTR encoded gene was. And it turns out that 3 groups described that there was an association with the T cell deletion function of minor lymphocyte stimulating antigens, and that the coding capacity segregated with endogenous copies of MMTV. So we haven't talked much at this meeting about endogenous viruses, but essentially all common inbred strains of mice have these. And so what the function of that is to stimulate lymphocytes to let them act as reservoirs for MMTV before the development of the mammary gland. Okay, so essentially what happens is you get infection of antigen-presenting cells in the gut, and then superantigen is put on the cell surface in conjunction with MHC class II, and then the C-terminus of superantigen interacts with the variable region of the beta chain of the T cell receptor. So whole classes of T cells, as opposed to 1 in 10,000 T cells, are stimulated, and they're stimulated to release cytokines, and they're stimulated for proliferation. So this allows the virus to survive in the lymphocyte reservoir, both B and T cells, to carry the virus to the mammary gland. Okay, so this was another collaboration with Susan Ross and also Tanya Golovkina. Here's a very young Tanya Golovkina right here. And so in collaboration with Susan, we had made LTR transgenic mice to try to figure out what, the function, or to prove what the function of the superantigen was in the MMTV life cycle. And so what we did was to express the LTR so that it was made in the thymus, so it would be considered a self-antigen. And because one of the effects of expressing superantigen to T cells is that eventually they become anergic, or they are deleted, they're lost from the peripheral circulation. And so we found if we expressed the exogenous MMTV superantigen in the thymus to get intrathymic deletion, then those SAG transgenic mice were resistant to infection. So this showed that you had to have that stimulation of T cells in order to create a susceptible mouse. Okay, so this is a report, early on by Craig Cohen and Harold about looking at these endogenous MTVs. And again, these are very useful to determine which sort of inbred strain you have, because the particular inbred strain has a certain constitution of endogenous viruses. So these endogenous copies, which are later called MTVs, italic, lowercase with a number, and these likely resulted from exogenous MTV proviral insertion into germ-line cells. Okay, so when we were trying to characterize the NRE and make essentially cloned isolate, recombinant isolates of MMTV, we would inoculate them into BALB/c mice, which are susceptible to the C3H strain. And oftentimes what happens is you get recombination with endogenous virus to correct any deleterious mutation. So my idea was, okay, we'll breed out the endogenous MTVs, and then we can analyze all of our viral mutants in these MTV null mice. So what we did is we bred them out, or Mary did this over a period of several years. And so by PCR, you can see that, BALB/c have 3 endogenous viruses, and the null mice had none. And as we expected, these null mice then have much higher levels of certain T cells that ordinarily would have been deleted by expression of the endogenous superantigen. But to our surprise, what happened was these mice became resistant to infection. So they were no longer able to be infected by exogenous MTV when they didn't have the endogenous MTVs. And we showed that with the type B leukemogenic virus, the T cell tropic MMTV. We showed it with exogenous MTV. It didn't matter whether it was milk-borne virus or injected virus, what age you infected them, they were resistant. So then this is why you should talk to your colleague. So my colleague in the lab next door works on the pathogenesis of bacterial pathogen, Vibrio cholerae and Shigella flexneri. And Shelley says, well, we can, maybe these mice are resistant to other sort of pathogens as well. And I said, oh, no, that won't work. And she said, well, we can find out in a week or two. So what you do is intragastrically feed the newborn mice with Vibrio cholerae and then do titers and determine what the lethal dose, 50 at 48 hours post-infection. And what we could find was, indeed, these were more resistant to a gram-negative bacteria, and that you could reconstitute that susceptibility by then breeding back any one of the individual MTVs. So I would say we still don't know for certain what the reason why these are resistant. But we do know from work by Tanya and Melissa Kane that MMTV transmission actually stimulates a lot of the immune system, so not only through SAG, but also through TLR4 signaling. So MMTV requires TLR4 signaling through the microbiota in order to get infections of pups. And if you don't have either TLR4 or IL-10, then you lose MTV transmission to pups through maternal milk. So because these MTV null mice are resistant to Vibrio cholerae within 48 hours, it must be some aspect of innate immunity that's defective. We actually can see the difference within about 24 hours. So that's too soon to have an adaptive immune response. And so our most recent result is that these MTV null mice, and what we did here is to take primary splenocytes from BALB/c mice and stimulate them with various ligands, and what we saw is that there is a differential response to CON-A, which we thought were just stimulating T cells, but actually in BALB/c you stimulate AID expression. But TLR4 is equally good. So we've done one more experiment, and it does seem as if one of the defects is in signaling through TLR2. Okay, so then I diverge to 2008. So this is a report from 4 of the full professors in my department. We sent a report to the scientists. We originally sent it to Science Magazine. They said this was old news. So what we noted was that when they were distributing merit raises in our department, and at that time there were no defined criteria for performance, more than $60,000 was distributed to the male full professor salaries, and the females received $750 total. And so what we did was threaten to sue the University of Texas for salary discrimination, and then subsequently they agreed to have a committee of unbiased faculty. They sanitized our CVs, and then they analyzed those, and they found that there was a direct correlation in the male salaries between accomplishment and salary. With women, it was an absolutely flat line. So this is not a pipeline problem. So discrimination is still rampant, it's insidious, and so if you see something, say something. So to one of the, another story about MMTV, and that's where even though there's a superantigen and there's a DUTPase, like in some primate lentiviruses, people still refer to MMTV as a simple retrovirus. So then, again, Melissa, who was a graduate student at the same time as Jenny Mertz, and together they identified a novel messenger RNA that a doubly spliced messenger RNA that was independent from the known GAG-PAL, ENV, and SAG singly spliced messenger RNAs. And when they sequenced this transcript, it turned out to be an in-frame fusion between parts of the surface protein and the transmembrane portion of the envelope gene. So it's very difficult actually to discriminate between or make mutations in this new gene, which we called REM, compared to the function of OMP. So what Jenny did was to devise a reporter assay, and we initially tried inserting parts of the MMTV genome into the HIV-based vectors that were being used to study REV. That never worked. But what Jenny did was to take the 3' end of the MMTV genome. She inserted the renilla luciferase gene in the envelope region. And really this was after Melissa had done a screen looking for where the REM gene might be functioning. And so this reporter gene was developed for what turned out to be the signal peptide function. So it really took a while for us to realize that the REM protein, which the original protein was 301 amino acids, but that if you blocked the putative signal peptide cleavage site with mutations, then essentially you didn't get cleavage either in the context of REM or OMF, and that you could normally, if you add back a REM expression vector, you could get very good expression in this reporter assay, but either of the signal peptidase mutations, it was absolutely dead. You can add micrograms and you never get a hint. So this is where we knew that it was really the signal peptide that was really responsible for a REV-like function. Okay, so then Ptolemy in the lab wanted to know, well, how can a signal peptide that's resident in the ER membrane go to the nucleus to bind to MMTV RNA and export them out for translation. And so she developed this retrotranslocation assay in which we put a biotin acceptor peptide at either the N-terminus or the C-terminus of signal peptide, and then we would express a ligase, a biotin ligase, in either the cytosol or the ER, and if you expressed the BAP tag on the C-terminus, you could actually get biotinylation of the signal peptide by virtue of adding streptavidin to get this gel shift. And Ptolemy also showed that when she made mutations to see which portions of the signal peptide were necessary for the retrotranslocation, that there were a couple of mutations between 57 and 61 in this 98 amino acid signal peptide that she could knock out the immunoprecipitation and interaction with P97 ATPase, which is known to be a chaperone protein that is needed for extraction of misfolded or misassembled proteins into the cytosol for proteolytic degradation. Okay, so this is really a cartoon summary of this. Probably many of you have seen this cartoon before, but essentially when you get REM, the doubly spliced REM messenger RNA synthesized, the signal peptide starts to emerge, and as the hydrophobic part at the C-terminus signal peptide is tethered in the membrane, then the C-terminal portion of REM gets glycosylated, and then it is somehow recognized as a substrate for retrotranslocation, which we still don't understand. And then, the signal peptidase will then cleave the signal peptide, which is tethered there, and then you get another protein, which we call REM-CT, for the REM-C terminus. And then we think that what happens is that instead of P97 recognizing a polyubiquinated protein and extracting it to deliver to the proteasome, P97 directly recognizes the SP portion, and then it is extracted, put into the cytosol, goes into the nucleolus to bind unspliced mRNA and shuttle it to the nucleus. And then because SP has an NES and an NLS, it can shuttle back and forth to take the messenger RNA, the unspliced messenger RNA out. Okay, so then this brings us to another story where murine apibec antagonizes MMTV replication in vivo. So many of you don't know that many of the studies that first described HIV VIF and its ability to antagonize the human apobecs, Susan and Kioma Okioma showed in mice, which have a single ApoBec3, that the MA3 null mice actually had very elevated levels of MMTV compared to the wild-type mice. So it appeared that this was the first in vivo evidence that MMTV replication in lymph nodes at early times in these wild-type B6 was inhibited by murine ApoBec3. And they also showed that MMTV virions package MA3 by binding the nucleocapsid protein. So then when we initially discovered that there was a C-terminal part, and we knew from Farah Mustafa's work that we could block the formation of that doubly spliced messenger RNA by making a mutation in the second spliced donor site. So they would make envelope messenger RNA, and they would make signal peptide in the Rev-like function just fine because they could make envelope. So this particular provirus, either an MMTV or a TBLV, which lack MMTV expression, actually have multiple mutations in both the WRC and TYC motifs. And so this is high throughput analysis of TBLV lymphomas. I actually did this experiment. And then Gurvani and Haywan have followed up by showing that there was an increase in WRC motifs, but only in the recombinant viruses. Again, I think that those viruses that are coming out of B cells are so mutated they have to recombine with the endogenous MTVs in order to have sufficient replication to infect the mammary gland. And then the TYCs, which are typical of MA3, are actually higher than what are found in the wild type. And so these WRCs, so this was true for both TBLV and MMTV, and that in this particular case, we looked at the RIM plus and RIM minus BALB-C tumors, and then we also derived mice by CRISPR, because most people do not engineer mice in BALB-C because it's very hard. And what we found is we lost those mutations that we thought were typical of AID. But the surprising thing is we also lost mutations in MA3. So there seems to be some sort of relationship between mutations, the presence of AID, and the ability to mutate the TYC. Okay. So, what is AID? So, many of you know that AID is activation-induced cytidine deaminase. It's probably the primordial ApoBec family member. It's involved in antibody diversity by both hypermutation of the variable regions of immunoglobulin genes, but it's also responsible for class switch recombination to go from IgM expressing cells to IgA or IgG expressing cells. So why is this AID important to MMTV? Well, it's been shown that MMTV requires replication in both T and B lymphocytes, again as a reservoir that is stimulated by superantigen to carry the virus from the milk and before the development of the mammary gland, so it serves as a reservoir to then transmit virus to the mammary gland. So then another student, Gervani, showed that REM actually induces proteasomal degradation of AID, so she could express either tagged or untagged forms of REM in the presence of murine AID tagged with GFP, and that in the presence of REM, that you would get greatly reduced levels of AID, and you could recover those levels in the presence of MG132. But if you did an experiment where you co-expressed MA3 in the presence of REM, there was no difference when you expressed various levels of REM. And then Gervani also showed and confirmed what Susan Ross's lab had shown, is that MMTV does package MA3, but it does not package AID. So the mechanism of inhibition must be different by these, for these two restriction factors. And then most recently, Polemy has shown that when we use an uncleavable form of REM, that you still get degradation of AID. So it appears that it's the full-length REM that is the antagonist of AID, and we think we've started efforts to identify the E3 ligase that's responsible for this degradation. So we think it is a cullen-based ligase, because if we use an inhibitor of the activation of essentially the E1 enzyme, then we get stabilization of AID levels. Okay, so this is an overview of what I've been telling you, is that MMTV is passed from mothers to pups in the milk. There's something like 10 to the 11th particles per mil of milk. And then MMTV infects gut-associated lymphocytes and also dendritic cells, and that these antigen-presenting cells put SAG on the surface, and then that superantigen is recognized by CD4-positive helper T cells that have the correct T cell receptors, and those are stimulated and interact with B cells. And then we get lymphocyte proliferation to infect more cells and to amplify the number of infected cells. And then after recombination with endogenous MTVs, we get infection of mammary epithelium, and then selection of cells with specific insertions to get mammary tumors. Okay, so then if the full-length REM is an AID antagonist and the signal peptide is a REV-like protein, then what is the function of REM-CT? So Wendy, at the time, we thought this was going to be the APOBEC antagonist, but Wendy characterized what was happening to REM-CT is that it was being cleaved and it doesn't have a transmembrane domain anymore. It is completely associated with membrane compartments, and she showed it's dependent on leaving the ER and going to the ergic via ARF1, ADP ribosylation factor, goes to the late endosomes that are RAB7 positive, and then using our old friend P97, goes to early endosomes, and her most recent data, which is unpublished, seems like a small amount of it ends up on the cell surface and then comes back in. We cannot detect it in exosomes or anywhere in cell supernatant. Okay, so we've also done some analysis by RNA-seq to look for what the function of REM-CT might be when we have eliminated the hypermutation induced by AID. So when we look at RNA-seq in B6 tumors, in the presence or absence of REM, we saw very little difference. But when we looked at B6 tumors, in the absence of REM and the absence of AID, we got hundreds of genes that were different, but only in the animals that were lacking AID expression. So when we did a Go analysis of that, it wasn't particularly helpful, but it does seem as if there is some signaling functions that are involved. And so we're still doing various purifications of the endosomal compartments and trying to decide where REM is going, or REM-CT is going, and what it's interacting with. Okay, so happy belated Mother's Day to those scientists who are mothers. This is my daughter, Dr. McLeod, and she's also a fan of aliens and Sigourney Weaver, and she sent me this: to the world's greatest mom. And then this is the card that my son, who is a structural engineer, sent me, and it says, too many things to do, too many places to be, not enough time for yourself, just screw it all. Okay, so instead of giving you a summary and listing all of the many people, I want to thank the many collaborators and students and postdocs who have contributed to this work. But instead of giving you that summary, I'm going to give you a poem that was written by me, not by ChatGPT. So through 50 years of reverse transcription, the Bittner virus defied description. Foster nursing showed one factor that gave mammary tumors faster. Hormones of the steroid ilk increase virus in the milk. These colloids, or particles type B, were maternally transmitted, you see, through guts and bugs with LPS that take their toll, but I digress. DCs show antigen that's not so super, to T than B cells, those party poopers. Activation is key, but what the heck, those lymphocytes make apobex. And to the rescue from mutation, RIM expression provides salvation. A hungry E3 eats some aid, though just how I haven't said. Virus rides lymphocytes to the breast, where Wnt and virus are suppressed. And NREs suppress transcription, then CUX1 cleavage saves the mission. A human rumor virus it may be, yet we still love our MTV.
Hi, Jackie. Thanks a lot for that. Could you expand a little more on the rumor virus? I mean,
you mean all Saul Spiegelman's work about saying the human virus was MMTV?
More recently, I think there's some papers coming out, and
there's papers constantly come out which I reject, but they get published anyway.
So what areas need still to be improved to maintain equity and diversity in science now?
I think, women have to step up and men have to participate too, and know that there is, you have to constantly look at the process for hiring, promotion, salary increases, just distribution of chair funds. All of those things have to be constantly monitored by faculty, and I think if students see that there is some sort of discrimination, then they need to find a friendly mentor, someone that's senior to go complain to.
You had a lot of female trainees.
Yes.
By looking at the pictures.
Yes.
Do you think it's just more appealing to work for a female PI?
We can talk about that over a beer.
That was a great talk. So in human, we know that some of these aprobic enzymes, when they upregulated, they actually leave a mutation signature in tumors. I wonder if in mice, people have shown any patterns of similar mutation in tumors.
Well, I just showed you that there are mutations in these tumors.
No.
You mean not in the provirus, but elsewhere?
Just in the tumor itself, not in the virus.
Right. I guess, yeah.
In the host genome.
Yeah. So we haven't extensively looked for that. We looked there, if you look at AID-induced mutations, it's previously been shown that AID does at probably 10 to 100-fold lower level cause mutations in certain genes like BCL6 and c-MYC. But, we did try to look for some of those, like in MYC and BCL, didn't see them.
Jackie, back here. It was interesting to see that the virus specifically targets dendritic cells. And
Yeah,
I guess
that's Susan Ross's work.
Right.
Yeah, that she did knockouts.
I guess that's part of the viral strategy because it wants to produce the superantigen in the antigen-presenting cells. But
right.
So dendritic cells, generally think of them as non-dividing cells. Does MTV have a way to infect non-dividing cells, or is it that the DCs do occasionally divide?
Yeah, I don't think that's really been carefully looked at, whether, but there is work by Hans Otto Arbeia where he said that they could infect non-dividing B cells. So I don't think it's been carefully looked at whether the virus has a way of entering through the nuclear pore.
Well, that might be of interest to look at.
Yep.
Hi, thanks for that great talk. Can you say a bit more about the endogenous viruses? Are they intact, or were they filled with APOE mutations? And
that's variable. In BALB/c, none of those those three endogenous viruses is replication competent. They all make a superantigen that is functional. So if we deleted any one of them, you could see the effect on T cell numbers. But there are strains of mice. The European strain GR actually has an exogenous replication competent virus that is in the germline. But I think over time those are selected against.
Susan, I'm sorry, Jackie, that was wonderful. So I wanted to ask you, so the virus has gone, it seems as though the virus has gone to a lot of trouble. It's established a relationship to be transmitted through the milk, but then it's got to involve a whole nother system to keep the transmission going. Is there any evidence that there is an advantage to the mouse of, in fact, just maintaining this relationship with this virus?
Yeah, I mean, so we did have, in order to create the MTV null mice, we actually got this strain from Jackson. We screened virtually every kind of mouse that they had to find one that didn't have endogenous MTVs. These mice breed like crazy, so they don't seem, at least in the laboratory, they don't have any disadvantage. Other than, my lab manager constantly complained because they could jump about 3 feet in the air. They acted like wild mice. So she would go like that to swing them by their tails to transfer them from one cage to another. Yeah.
Hey, Jaquelin. Here.
Oh.
Yeah, that was an amazing talk. So you talked about the endogenous retroviruses in the inbred mouse, but I wonder if they can also be found in dirty mice or pet store mice, and since those mice are more
by and
likely
large, yes,
to find other viruses, are they protected in some way from other virus infections?
Yeah, I think, this work has shown pretty clearly that the endogenous viruses are skewing the T cell repertoire. So I've had many, many requests for this from these mice for study by immunologists. So, we're getting ready to ship them to UCSF for people that are looking at autoimmune responses. Like I said, you can find mice in the wild, those para-attack mice that I was talking about that we use to generate the MTV null. They were recently inbred from wild. That's why they are really not well behaved. But yeah, you can go out and catch mice, and most of them will have endogenous MTVs. But there are some that will lack it.
I have a conceptual question about the generation of these MTV null mice. You mentioned they were bred over years. Nowadays we have other methods, so if you...
Well, you must look at the time scale of those publications. CRISPR was not available at the time.
So nowadays it would be possible to create these with CRISPR, or are there any other restricting factors that would prevent us?
Yes.
To do? Okay.
So wonderful talk. So I have a curious question. So you show that the it was a viral protein or the virus itself that goes to ERGIC and then the Rep7, Rep5 and the
That's a viral protein. REMCT goes from the ER into the ERGIC to late endosomes, early endosomes, and then the cell surface.
And you did not see any protein in the exosomes, right?
We did not. We did not find it anywhere extracellularly.
So is there any possibility that the protein is, transported from one cell to another cell by physically connected cells?
Yeah, we couldn't rule that out. Yeah, we haven't looked at that.
I see.
Hi. Do you know anything about the origins of the superantigen? Is there any signature going back? Is it derived from something?
Right. So, if you look at the most closely related, human virus, which would be HERV-K, those do not have any superantigen-like genes. So it's definitely been picked up. Presumably it had something to do, well, I guess Bridget Huber had some speculation about how it has to do, they may be part of the class 2 MHC presentation pathway that they've co-opted.
Thank you very much. I think no one believes anymore in simple retroviruses. It's really complicated.